Hello, everyone. Welcome to the American Thoracic Society's Journal Club, jointly organized by the Allergy, Immunology, and Inflammation Assembly and the section on Genetics and Genomics. Today, we will dive into a recently published European Respiratory Journal paper titled Extensive Acute and Sustained Changes to Neutrophil Proteomes Post-SARS-CoV-2 Infection. The goal of our journal club is not only to stay updated with the current research in proteomics and respiratory diseases, but also to sharpen our critically thinking skills and collaborate as a community of learners. First of all, it's my great honor to invite the authors of this paper, Dr. Merete Long and Professor James Chambers, and also three prestigious panelists, Professor Dong Xin, Dr. Theodoros Kapelos, and Dr. Jezreel Panteleon to this journal club. Dr. Merete Long is currently a research fellow at the University of Dundee with expertise in the translational research for respiratory infections, such as COVID-19 and bronchitis. Professor James Chambers is a professor at the University of Dundee and Asthma and the Long UK Chair of Respiratory Research. James is also a world-renowned bronchitis expert. And as many of you know, he is also the chief editor of the European Respiratory Journal. Professor Dong Xin is the director of the Center for Heart, Lung, and Innovation, and a professor of medicine at the University of British Columbia. Dong is also a world-renowned COPD expert. He has served on the GOLD Scientific Committee since 2009, and as many of you know, he's also the deputy chief editor of the European Respiratory Journal. Dr. Theodoros Kapelos is a junior group leader at the Institute for Lung Health and Immunity at Hamalos Central Mutual. He also has the expertise in how immune cells contributed to the progression of obstructive airway diseases. Dr. Panteleon is a postdoc fellow in the pulmonary medicine at the University of Texas MD Anderson Cancer Center. He has expertise in the development of molecular cell and system biology approaches to understanding the mechanisms of respiratory infections. Lastly, myself, my name is Zhaozhong Zhu. I'm the assistant professor at Mass General Hospital and Harvard Medical School. I'm also the director of Translational Research Unit in the Emergency Medicine Network at MGH and Harvard. So after the introduction, to start us off, we will have Dr. Long present this paper for 25 to 30 minutes, and then we will have the three panelists comment on and discuss the paper with the authors. Lastly, we will welcome the audience to share any additional comments and questions they may have. So thank you so much for being here and bringing your curiosity and expertise to this discussion. And let's dive in. Dr. Long, please feel free to take over. Thank you.
Thank you very much. So first, thank you so much for the opportunity to share our work with everyone today and hopefully generate some interesting discussions. So I'll dive straight in with a very small introduction to our favorite innate immune cell, the neutrophil. These cells develop in a multi-step maturation process in the bone marrow, and amongst other things, they sequentially develop their different granule types. The mature neutrophil is then released into the circulation and patrols around until it encounters an inflammatory stimulus or inflammatory cue. It then migrates towards the source of this chemoattractant in the tissue, and in the case of COVID-19, that would of course be the lung tissue. So neutrophils have several key effector functions when they arrive, including things like phagocytosis to remove invading pathogens, the release of reactive oxygen species, as well as release of their granule content via degranulation. And the granules, of course, having quite high levels of neutrophil proteases, capable of antimicrobial activity, but also quite capable of tissue damage. Neutrophils can also release web of their DNA and proteins in a process called netosis. And quite importantly, and increasingly importantly, we found in COVID-19 research, these cells can talk to other immune cells, and they have immunomodulatory capacity for both adaptive and innate immune responses. So as a very brief overview of research into neutrophil biology, these cells were discovered just under 200 years ago by Paul Ehrlich, and this was done using his newly developed histological staining technique. And research has slowly progressed into understanding more and more about neutrophil functions, from first phagocytosis to netosis actually relatively recently, just over 20 years ago, and with their immunoregulatory capacity being most recently discovered. And omics technologies and high resolution techniques are really driving the way forwards in the last couple of years into neutrophil research and allowing us to really understand the previously underappreciated heterogeneity and diversity of these cell types. So in COVID-19, what we know about the immune response in severe disease was firstly that there was lymphopenia, so a reduction in T cells and B cells, that there was T cell activation, but there was also T cell dysfunction and exhaustion. There were different kinetics and magnitude of antibody production associated with severe disease, as well as cytokine storm, so the release of cytokines en masse, creating a hyperinflammatory response. And the first thing to be noticed, perhaps about neutrophils, was that they were increased in numbers in the circulation in severe disease. And perhaps slightly unusual for an acute respiratory infection, it was identified that these cells are having activity in the circulation. So they were releasing their granule products, including neutrophil elastase, and higher levels of circulated elastase were associated with adverse outcomes in COVID. And this included things like ICU admission, as well as respiratory failure. Equally, nets or neutrophil extracellular traps were also being released into the circulation, shown here by the histone DNA complexes. And these were equally associated with adverse outcomes. And one of the adverse mechanisms in COVID-19 resulting from release of nets into the circulation was the formation of microthrombi, so aggregation of platelets in the lung vasculature. And this is a fatal case of COVID-19, shown here by immunofluorescence imaging of the lung autopsy specimen. And nets are shown in green and white, and a platelet aggregate is indicated by the cluster of red cells. So one of the most, informative early studies using these kind of high resolution techniques to profile the immune response was this study from a group in Germany published in 2020. And one of our panelists is a co-author for this paper. So this group used a combination of different high resolution techniques, including mass cytometry, single-cell RNA-seq. And they looked in detail at the peripheral blood immune response in COVID in mild and severe disease. And they found that in severe disease, neutrophils were different. And amongst other things, there were more immature neutrophils released due to emergency myelopoiesis. And these neutrophils also indicated that they had increased immunosuppressive capacity and had increased expression of things like programmed cell death ligand 1 in severe disease. Then, utilizing proteomics, this relatively small but quite important study came out in 2021, which used proteomics to profile peripheral blood neutrophils in a control cohort and in patients who had severe COVID-19 who developed acute respiratory distress syndrome. So even in this small study, these authors were able to profile neutrophils using proteomics and identify that there was a really high magnitude of differences in severe COVID-19. And this included things like enhanced interferon signature in COVID, as well as altered antigen presentation capacity and altered metabolic activity. So this is kind of where our study fits in. So there had been no systematic analysis of neutrophil proteomes that had explored the relationship with a range of severities and also longitudinally in recovery or non-recovery, as the case may be. So to address this, we designed Predict COVID-19 with the aim to do exactly that, to profile the neutrophil proteome in COVID to understand immune responses in acute disease across a range of severities and also during recovery. So for this, hospitalised patients were enrolled at Nine Wells Hospital in Dundee in Scotland into one of three groups. So we had a COVID-19 group, a control group who were hospitalised patients that had no known infection, and a non-COVID lower respiratory tract infection group. And we enrolled patients from May to December 2020. So each of these participants at baseline had a blood sample taken, and just in the COVID-19 group, we also took longitudinal samples, including at day 7 and day 29. So for this work, we actually really quickly set up at the beginning of 2020 our decommissioned category 3 containment lab to be able to process these samples. So we isolated from the fresh blood samples, peripheral blood neutrophils, and we did this using immunomagnetic separation. And we use negative selection. And so the result of this is that we get a relatively untouched neutrophil population that's quite pure. After we'd stored quite large batches of samples, so between 50 and 100 samples, these were given to our study colleagues and collaborators at Life Sciences who process the samples for mass spec and then run this analysis. And we use untargeted proteomics, so this was operated in data independent acquisition modes. And our collaborators also further performed bioinformatics analysis, allowing a comprehensive understanding and overview of the data. And to give you an idea of the scale of this, we were able to identify around 4,900 unique proteins per sample. So we had a lot to look at. So first, regarding the characteristics of our study participants, the COVID cohort was quite representative of those published in clinical studies and trials at the time. So our COVID group tended to be older individuals, and they did have some cardiac and respiratory disease comorbidities, but these were well matched in our control group. The majority of our participants required some form of supplementary oxygen, with around 20% actually requiring high flow oxygen or ventilatory supports. So we first wanted to take a global overview of these changes, and we compared our respiratory infection groups, both COVID and non-COVID, to our control neutrophil proteomes. And looking first at our non-COVID lower respiratory tract infection group, there are over 400 proteins that were differentially abundant in the neutrophil proteome compared with controls. But when we compared our COVID-19 proteomes to controls, you can see that the magnitude of these differences is much, much greater. So there were over 1,700 differentially abundant proteins in the COVID-19 proteome. We looked for proteins that indicated a kind of universal COVID-19 signature. And as had been shown by others, we saw a really strong type 1 interferon response in the neutrophils. And quite interestingly, this allowed us to actually identify individuals that were asymptomatic or pre-symptomatic that had originally been enrolled into our control group. So MX2 was one of the most highly expressed and differentially abundant proteins in the interferon response, which is an antiviral protein regulating nuclear important viral replication complexes. And these two individuals were subsequently moved to the COVID group for further analysis when we finalized our data set. And overall, just over 100 proteins were part of this interferon signature. When we stratified our participants with COVID-19 by severity, so for all of our analysis, we used the WHO 7-point ordinal scale, and this was the gold standard for reporting COVID severity at the time. So WHO 3 represents participants who were hospitalized but didn't require supplementary oxygen. WHO 4 were participants who did require supplementary oxygen, but this was via face mask or nasal prongs. And WHO 5 to 6 are our most severe participants who required the high flow oxygen or ventilatory support. And we had increasing levels of differences with increasing severity. However, many of these changes overlapped. It was just that in severe disease, we see more and more proteins that are differentially abundant. So looking back a little bit more at our COVID-19 signature, regardless of severity, and proteins that were downregulated, we actually see 70 proteins linked to nuclear rigidity. And these included things like SINE1, SINE2, and SUN2, and these are all part of the link complex. So this links the nuclear skeleton and the cytoskeleton. And link provides a bridge between actin and things like the lamin B receptor, which sits on the inside of the nuclear membrane. And overall, these proteins are able to regulate nuclear stability and nuclear shape. So a reduction of these potentially means that the cell has greater plasticity for things potentially like cell migration. We then looked at changes that were associated with severe disease to understand what's really different about these patients and what inflammatory processes might be driving severity. And we first found several proteins that were related to metabolic processes were altered in the most severe individuals. So for those that were reduced in abundance in severe disease, we found that glycogen synthase kinase, or GSK3A and B, were both reduced, and these are inhibitors of glycogen synthesis. So less inhibition may mean more storage of glycogen in the cell. And we also saw that there were reduced levels in more severe individuals of PYGL1, which is glycogen phosphorylase involved in glycogen breakdown. However, when we group the proteins identified across metabolic processes together and have a look at severe disease, we can actually see that this regulation of glycolysis is the only process to be downregulated. And we see processes including glycolysis, fatty acid oxidation, electron transport chain, and the Krebs cycle are all increased in severe disease. And we know that some of these processes, including fatty acid oxidation, Krebs cycle, are higher in immature neutrophils, so it may be associated with the immature signature that other authors have also shown. We also saw differences other than this metabolic change in severe disease, and this included things like increases in cell signaling receptors and surface receptors. So TLR2, or toll-like receptor 2, was increased in severe disease. This is a pathogen recognition receptor, which once activated initiates inflammatory signaling cascades, including via NF-κB. We saw an increase in VISTA, which is an immunoregulatory receptor, and it's capable of suppressing T cell activity. And we saw actually increases in several cell surface receptors that might link in with this kind of hyper-inflammatory profile and the ability of the cells to quickly respond to inflammation. Interestingly, we saw a decrease in the total estimated protein content of just one granule type in the severe group, and this was the specific granules or secondary granules for which the defining protein is lactoferrin, which might indicate a degranulation response. And we also saw an increase, as other authors have now shown as well, and in the introductory studies that I showed, this was one of the key findings, that arginase 1 was increased. So this is able to degrade extracellular arginine, which is essential for T cell survival, metabolism, and proliferation, and is potentially capable of downregulating T cell activation. So in acute disease and in severe individuals, we see upregulation of many cell surface receptors that would be capable of triggering inflammatory responses. We see an upregulation of potential immunosuppressive capacity of these neutrophils, a downregulation of glycogenolysis proteins, but an upregulation of many other metabolic pathways, including glycolysis. And we see indications of degranulation, at least for specific granule types. So we also had longitudinal samples in our study, and that was something that we were really interested to understand, was the kinetics of the inflammatory response and the neutrophil response. So when we had a look at our longitudinal samples together with the baseline samples, we could see in the PCA plot that the day 1 samples from our most severe individuals tended to cluster separately. So these are most of the samples shown within the orange circle. However, there were differences longitudinally. So our day 7 samples tended to be found in the bottom right-hand side of this plot, and our day 29 samples at the end of our study tended to be found over on the right-hand side. So there did seem to be a progressive continuum of proteomic differences. And this was associated with disease severity. So when we compare the baseline status or baseline severity of these patients and compare the differences between day 1 and day 7, we see that the milder patients actually had very little differences 1 week post-enrollment. However, the more severe participants had over 1,000 proteins that were differentially abundant. So some things were increased at day 1 and remained unchanged, including the potential immunosuppressive capacity of our neutrophils. And sorry, maybe I'll explain a little bit about the graphs, but the controls are in pink on the very left-hand side. Our day 1 samples are in light orange, and our day 7 samples are in dark orange, and all patients are stratified only by their day 1 severity. We also saw things that were totally changed from day 1 to 7 in our most severe individuals. So, for example, GSK3b was reduced at baseline and was conversely increased at day 7. And we saw unique changes, including upregulation of inflammatory cell receptors, including FPR1 for formulated peptide receptor 1, which recognizes peptides from microorganisms as well as host organisms to trigger inflammatory responses and migration of the cell. And interestingly, we saw an increase in the IL-1 receptor 2 decoy receptor. And this is typically suppressed by inflammatory stimuli, including LPS and interferon gamma. So it was an interesting increase here at day 7. So we also looked at changes that were specific to severe individuals that were downregulated or reduced in abundance by day 7. And pathway analysis indicated quite a strong type 1 interferon signature where type 1 interferon signaling might be reduced. So these proteins were grouped together, and we see at day 1 in our milder individuals, there's quite a robust interferon response induced. And at day 7 in these milder individuals, this is maintained. However, when we start to look at our more severe participants, the difference from day 1 to day 7 is quite stark, where we have a significant reduction in most of the interferon signature just 1 week later. And this is perhaps most pronounced in those most severe individuals. So we have a loss of type 1 interferon signature at day 7, 1 week after enrollment in severe disease. There may be many explanations for this. However, it may be partly explained by significant reductions in regulators of type 1 interferon proteins, and this includes CMTR1, which was reduced at day 7 in severe individuals, and is a transcriptional regulator of several interferon proteins, including MX1, which was also really significantly reduced. We see upregulation or potential for upregulation of other inflammatory pathways, including NF-kappa B signaling. So we had a significant reduction in NF-kappa B inhibitors, including NF-kappa B inhibitor epsilon, shown here. This is usually able to trap NF-kappa B in the cytoplasm, preventing it from going to the nucleus and performing its activity as a transcription factor. And we also see that histones, and particularly H1 linker histones, are reduced at day 7. So these sit outside the nucleosome beads, keeping the DNA in place. And again, this potentially indicates more plasticity of the chromatin and may link in with potential phagocytosis. So at day 7, we had a sustained increase in several receptors controlling the immune response, a sustained immunosuppressive signal. In addition, we have an increase in migratory receptors. We had the sustained decrease in glycogenolysis related proteins, a sustained increase in glycolytic proteins, and I didn't show this as part of the day 7 data, but we also see sustained degranulation signature. So we, in addition to proteomics in the study, also performed some other analysis that didn't make it into the paper, but we analysed a little bit of the function of the neutrophils at day 29, so the last time point that we had in the study. And when we looked at phagocytic capacity of these cells, we saw that there were overall changes from day 1 to day 29, where phagocytic ability actually seemed to be less at day 1. We also performed mRNA-seq of whole blood leukocytes, and we found that when we analysed these samples at day 29 and split participants by recovery or non-recovery, and the difference in these patients is actually quite fine. So the WHO1 participants had been discharged home from hospital and reported that they had no limitations on daily activities, whilst WHO2 individuals had also been discharged from hospital, but self-reported that they did have daily limitation on activity after COVID-19 infection. And we quite interestingly saw a neutrophil gene expression signature, which might be representative of the persistence of immature cells, or might be representative of changes in neutrophil activity. We also measured neutrophil granule products in serum, and we found that in the non-recovered participants at day 29, we had high levels of things like azurecidin, which is contained in the primary or azurophilic granules. And in a quite simple assay, we measured degranulation. So we took whole blood, stimulated these cells, and then collected the plasma, also allowing us to measure degranulation products there. And we actually found that we weren't able to stimulate these cells, whether individuals were recovered or not recovered, to produce any more elastase. However, quite interestingly, when we look at vehicle control conditions, so the unstimulated cells, the non-recovered individuals seem to have higher levels of elastase being released. So then we used our proteomic data to answer the question of, are these neutrophils really different? And the answer was yes. So in recovered individuals, there were just over 300 proteins that were differentially abundant at day 29 from our control group. But there were many more proteins that were significantly altered in non-recovery. So the neutrophil proteome in non-recovery was really highly different to our control group. Many of these proteins changed in recovery and in non-recovery at day 29 tended to overlap. So first we looked at those which might just indicate the overall COVID-19 signature and might just be associated with post-infective processes. And this included Fc receptor gamma, which was reduced in abundance. And these graphs are indicating controls in the top in pink. And we have data from each of the time points collected. So the day 1 samples are in blue, day 7 are in yellow, and day 29 are in grey. And the non-recovered individuals at each time point are indicated in the darker colour of each set. So the reduction in FcR gamma may indicate potentially a reduced ability to recognise IG-opsonized pathogens. We had a sustained reduction in PYGL, which is involved in glycogen breakdown. And we also had reductions in migratory receptors in both recovered and non-recovered individuals, including the leukotriene B4 receptor, as well as an increase in receptors including CF2RA, which was potentially involved in activating pro-survival signals after CSF stimulation in these cells. And in both non-recovery and recovery, we saw indications of our degranulation signature. So the specific or secondary granule proteins were reduced in these cells. And in addition, something that we hadn't seen at other time points was that the estimated protein content of the phycalin granules, the fourth granule type, were also reduced. So lastly, things that were specific to non-recovery. We found that several proteins relating to glycolysis and modulating this process were actually reduced. And this included SLC2A3, which is GLUT3, so a glucose transporter that brings in glucose from outside of the cell. And there were actually several key glycolytic mediators that were systematically reduced. So we think that we have a reduced glycolytic potential in non-recovery at day 29 that wasn't seen at any of the other study time points. We also saw indications only in non-recovery of persistence of immature neutrophils, shown here by a reduction in CD10 abundance, as well as increased indications of cell activation. So CD64 was increased in non-recovery, and this is an activation marker, an FC receptor, and it's actually been identified as quite a sensitive marker of infection and sepsis on blood neutrophils. In addition, we had reduction in several integrins, including CD11B, which facilitates cell adhesion and migration to tissues, as well as several other cell surface receptors associated with cell migration. And finally, we had a reduction in proteins or inhibitory proteins, including SHP1 and SHP1. And both of these are able to negatively regulate cell survival and activation. So reduced levels of proteins like SHP1 are associated with autoinflammatory conditions like IBD. So in conclusion, at day 29 in non-recovered individuals, we had a reduction in inhibitory receptors, a reduction in migratory receptors and integrins, reduced granule proteins indicating degranulation, a reduction in glycolytic proteins, as well as a sustained reduction in glycogenolysis. So putting these all together at day 1, we had neutrophils, which may be able to more rapidly respond to inflammatory stimuli, capable of immunosuppression, potentially degranulating, but with increased metabolic demands. We also showed that in severe disease, the interferon signature was rapidly lost, and we saw this by day 7. At day 7, many of these changes were sustained, in addition to an increase in migratory receptors. And at day 29, we had a reduction in inhibitory potential of these cells, indicating that there may be more ongoing inflammation, which is less able to control, a lower ability of the cells to migrate, and impaired metabolic activity of these cells, as well as enhanced degranulation. So I hope I thought maybe have a few minutes just to go through very briefly some study limitations and areas for potential further work. So this study, by its nature, is descriptive, and there are key mechanistic experiments to follow up on. The inclusion, of course, of autoimmune cells would be great in future studies to understand the impact of our findings in the lung tissue. Because we recruited from May to December 2020, our data represents patients from the first and second waves of the UK COVID-19 pandemic, and of course this was performed at a single centre in hospitalised patients. Vaccination has changed the profile of severe COVID-19 in hospitals, and further investigations of the impact of this would be of interest. A longer term follow-up would of course be of interest, and some of these things we've started to address. So proteomics in our study allowed us to identify closer to 5,000 unique proteins in neutrophils from healthy individuals during acute infection, during recovery. In severe acute disease, we had emergency myelopoiesis, metabolic changes, potential functional changes, immunoregulatory capacity of these cells was altered, and we saw indications of hyperactivation. And delayed recovery was associated with the persistence of immature cells, even up to day 29, post-enrollment, degranulation, a reduced potential for inhibition and migration, and altered metabolic profiles. And this leaves us with the questions of, does this mean that these individuals might be at increased risk for systemic harm and secondary infection? Or is this potentially an intentional neutrophil disarming technique that's employed to counteract the hyperinflammatory responses encountered in acute severe disease? And we've used neutrophil proteomics to characterize now several other diseases in studies quite similar to predict COVID-19, including influenza. And we've also followed up patients with post-COVID syndrome for 3 to 12 months. And use these techniques within clinical trials to be able to understand the effect of investigational treatments. And we've also embedded them into the future trials, repurposing anti-inflammatory therapies in bronchiectasis. And with that, I would like to say thank you very much for everyone involved, to everyone involved in the study. It was a really huge team effort from a lot of different groups. And thank you to the Tayside Clinical Trials Unit and Clinical Research Centre who facilitated the study. And thank you to our participants and their friends and family who supported them to participate, and to, of course, the funders for supporting this work. So thank you very much, and I'm happy to take any questions.
Great. Thank you so much, Dr. Long, for the fantastic presentation. Personally, I have learned a lot. And this is very, very important study, focus on the neutrophils for proteomics analysis, given the importance of neutrophils, as you mentioned, in the respiratory infection and COVID-19. And as we know, many of the omics study can only rely on the bulk specimen types, but not on the more granular and specific cell types. So the omics signal can be heterogeneous and can be hard to capture the very detailed mechanisms. So this study is truly fantastic. So now I want to welcome the 3 panelists, Professor Singh, Dr. Kapelos, and Dr. Pantaleon, to join the discussion for their comments and questions. And then we will we see some questions from the audience, that we will go with the audience. So maybe we can start with Professor Singh.
Well, thank you, Zizi, and thank you, Merete, for an excellent presentation, and it's a lovely paper. ERJ is obviously proud to publish it. So I had a couple of comments and a question. So, it's a shame that you didn't get tissue-specific neutrophils to figure out exactly what's happening. We've done it in long COVID, taking bronchoscopies and bronchial brushes, and there is still persistent neutrophilic signature, although the number of neutrophils in the small airways is quite scant, but it's definitely there, signatures there. So among people who have long pulmonary COVID. So I think there is an inherent neutrophilic issue with COVID, and probably persists into the long COVID. The question I had was whether you think the signature that you found in the circulating blood, both in the acute and the chronic setting, reflects an inherent problem of the myeloid lineage. So in other words, it's a stem cell or some sort of progenitor cell dysfunction that is unearthed by COVID, what was unearthed by COVID, or do you think this is just a reactive process to quote unquote the sickness of the individual, although maybe contributing to the sickness? So do you have any thoughts about that and how you could tease that out going forward?
Yeah, it's a great question. So in terms of innate or inherent dysfunction in individuals, I think that since we see that in recovery, there is an increasing tendency to be similar to our control neutrophils, it would indicate, at least in individuals that are severe enough to be hospitalized, but don't end up needing, for example, high flow oxygen. This doesn't seem to be an innate defect. And I think if we had a look at neutrophils, for example, at 3 months, we would see that these are really the same as the controls. And I think that we would see more differences between our control population and our recovered individuals if this was an innate defect. In those who do go on to have the most severe disease, I guess that is potentially more interesting. So in terms of how we figure that out, well, we do have some ongoing studies, or rather James has some ongoing studies to have a look at the genetic background of these patients to understand if there is links with the genetic background and cell function, since we did a lot of other work to profile the function of the cells as well. So that may provide some answers to whether there are innate differences. But I think it's likely other potential environmental factors. We didn't see big differences or significant differences in really any of the clinical parameters between recovered and non-recovered individuals. So they didn't have high levels of chronic cardiac disease, chronic respiratory disease. And I think if it was innate, these individuals would also be more predisposed to chronic conditions, and they don't seem to be. So I think the answer is no for now, but it needs a lot of further work.
James, I think you wanted to chime in?
Yeah, I was just, I mean, I agree with everything that Merete has said, just on the innate versus what's driving the changes at day 29. In other cohorts, we have done some proteomic analysis of plasma, for example, in people with long COVID, and found a lot of elevated inflammatory markers, including IL-6. And some of the changes that you see in these neutrophils, you know, the metabolic exhaustion at day 29 and the reduction of CD11b is things that you see in other chronic inflammatory diseases. So I would, your question is an invitation to speculate, and I would speculate this would fit with the idea that some patients transition to a more chronic inflammatory state rather than it being an innate myeloid defect.
And I just have a follow-up question to that. So your top hit seems to be in the pathway of metabolism. That sort of raises the issue of whether the mitochondrial function has been perturbed in some way. Did you look at the mitochondrial function of these neutrophils in the sick and the long COVID people to see whether there's an inherent mitochondrial dysfunction?
Yeah, so we haven't done any specific analysis looking at mitochondrial function. Obviously, some of the pathways that were particularly upregulated in acute disease are associated with mitochondrial metabolism, so fatty acid oxidation and TCA cycle, electron transport chain. But we know that immature neutrophils tend to have a higher reliance on these kind of mitochondrial pathways. So what we actually think is that this may be because we have many more immature neutrophils present in severe disease, and so their metabolism is different. But yeah, it would be, of course, nice to do and nice to understand a little bit more.
Thank you, Dr. Long. I see Jess is raising his hand. Jess, please.
Yes, great work again. It's kind of like a related question to Dr. Sin's question, and I was wondering if, do you see those same changes in day 1 or day 29 between the COVID cohorts and the other respiratory infection cohorts? Because I think that comparison is actually a good one to help you tease out whether these defects come from the leukocytic or the innate defects, or if it's a pathogen-specific marks. So I would like to hear more about that comparison between more COVID versus non-COVID cohorts, please. Thank you.
Yeah. So we didn't have longitudinal analysis for our non-COVID groups, which would be really nice to do, and I think is kind of an under-researched area at the moment for non-COVID respiratory infections. But we did compare, of course, the baseline samples in LRTI and COVID-19. So we obviously saw a much higher magnitude of kind of inflammation, interferon signaling. The majority of pathogens identified in our LRTI cohort were bacterial. there was just one of those that had a virus recorded as the causative pathogen, although the majority were actually unknown. So I think some of the differences there probably lie in the fact that we're actually comparing potentially bacterial infection with a viral infection. So we didn't have an interference signature. We had much less involvement of kind of complement. And interestingly, when we compare the two, I think there was a lesser element of neutrophilic inflammation in LRTI. So we had lower levels of elastase, for example, in the LRTI group compared with COVID-19.
Thank you, Dr. Long.
To add to that, one of the complexities with the LRTI group is just how heterogeneous they are. And I think if we had the opportunity to do the study again, doing a like-for-like comparison with something like influenza that's well characterized might make it more clear.
Yes, I agree with James. Thank you, James. Dr. Theodoros-Kapelos, do you have any comments or questions?
Yeah, sure. First of all, let me apologize for the fact that my camera is not working. I tried to fix it earlier, but it wasn't possible. But Merete, really nice study. And what I noticed is that every figure was almost, it could be a sub story on its own. There's actually many little nice messages that for me, two of the most exciting ones were first of all, the metabolic analysis of the neutrophils and the changes that you saw during but in the more severe cases of COVID, particularly the increase of the glycolysis-related genes and fatty acid oxidation, which is something that I have also seen in other diseases than COVID. So especially chronic diseases, the one that you are focusing on with James, but also COPD. And I was really curious because there's not really much known about neutrophil metabolism, and I was wondering whether how do you reconcile these different pathways being so differently regulated at the same time? Have you perhaps looked at it, or maybe like James said, can I invite you to speculate?
Do you mean between acute disease and recovery, or the glycogenolysis produced while something else is
the different stages, the WHO 2-3 versus 5-6. I saw the difference was more striking there.
Yeah, so as we talked about a little bit, I think it definitely has something to do in acute disease with the presence of the immature cells, which have a different metabolic profile. And I think in terms of the link with non-recovery, I think as James mentioned earlier as well, that this starts to look a little bit like what we see in chronic lung disease. So for example, in COPD, so during an inflammatory response, in theory, the neutrophil relies on glycolysis, and it also needs to both build and use its glycogen stores, especially in hypoxic conditions. But what we see in non-recovery is a reduction in both of those things, and that is something quite similar to what's seen in COPD, where the cell then is potentially not able to meet its metabolic demands, and that results in some of the functional changes that we see in kind of a reduction in phagocytosis and altered cell survival. So I think a lot of the changes do make sense. It's just something that we hadn't necessarily seen in acute infection before, and I think there's quite a lot to be investigated overall for neutrophil metabolism, and to really understand what processes are really employed in disease and in health, and the differences between in vitro experiments, of course, and the real case in vivo, which this is hopefully helping us to unravel. And you mentioned that there were similar changes in bronchiectasis that you'd seen as well. Is that right?
We've seen it now in our published data that we have. So it's always interesting to see that the fatty acid oxidation is usually elevated in disease now, acute COVID as well, and it goes hand in hand with glycolysis. So I remember in your discussion, you made the point that is not that probably is that not all the consequences that you see in the marker changes or the functional test that you tested a result of the metabolic changes. So you think that perhaps is something even more upstream that changes both metabolism and the other functions, or could it be a story of a chicken and the egg? So they all happen simultaneously, and it's really difficult to pin the point.
Yeah, it's a great question. I honestly don't really know. We see quite direct kind of upstream regulators. So, for example, PYGL being changed and GBE1 and glycogenin, especially involved in the changes that we see in glycogenolysis. But in terms of what's upstream of that or what's actually causing that, I don't know. But yeah, if anyone else wants to speculate, that would be great.
Thank you. And I see Jess has raised his hand again.
Yes. Just one quick question and one quick comment. First comment is it's really nice to see such extensive and carefully done descriptive omics studies from clinical databases. I don't think it's necessarily a limitation. I think at the stage where we are in integrating omics-based technologies into clinical research and clinical application, it's actually an advancement that we're able to get so much detailed information. So congratulations on that. And also, I'm looking forward to see what your work from the transcriptome of the neutrophils is going to go. Do you know whether that's going to be used to describe the baseline profiles with COVID, or is it an approach mostly focused on the longitudinal profiles? And how would you integrate that with your current proteomics?
Yeah, so we haven't, I mean, I would have loved to, but we haven't performed neutrophil transcriptomics in our pure cell population in the isolated cells. So we did this in kind of all peripheral blood leukocytes using the PAX gene tubes, and there is some published work on this, and I think also longitudinally showing those kinds of changes. But it would be really nice to link this up with the proteomics data, since of course we know for neutrophils there is a little bit of a mismatch between the protein level changes and the neutrophil transcriptome. So it would be really nice to try to pull that out and do some data deconvolution for our transcriptomics to kind of imply a little bit more specifically about the neutrophils. In terms of how to actually do that, I think there are people much cleverer than me that are able to do that bioinformatically. But yeah, it would be a great next step to do, and we're hoping to do these kinds of studies in the chronic lung diseases that our lab kind of typically studies, like bronchiectasis.
Don, and then James.
Well, you know, now that you've done the study, if you had to do the study again, hopefully there is not again COVID. It's a once in a generation and once in 100-year thing. Would you choose a unbiased platform like MSMS, or would you be much more targeted, you know, and much more quantitative, like flow or CyTOF or spectral flow now? So, you know, I think it's fine for the first study to be like this, but I think there's a lot of nuances that are being missed because of the platform that you use. Do you feel that way, or do you think MSMS is still very good for exploring, you know, mechanisms?
Yeah, I think you're right. Definitely a little bit of everything is great. So for example, the study that Dr. Capellas was involved in used different kind of high resolution platforms to profile both protein level and transcriptome level. So I think proteomics, I would definitely include. It allows us to look at thousands and thousands of things and discover things that we wouldn't have even thought to look at. But I think in combination with things like mass cytometry, like spectral flow, and single-cell RNA-seq is definitely the way to go, and that's how we've started to design the kind of upcoming trials and observational studies that we're doing in chronic lung disease. So yeah, definitely a little bit of everything is better.
Yeah. On the positive side, there is extraordinary power in being able to profile 4,900 proteins in a single shot, and being able to embed it within a clinical study, as Jez said. So I think there is power in this platform, but and we never want to do this again, Don. But the future is multi-omic integration, isn't it? So the future is putting together proteomics with functional data, and being able to learn more about what the implications of the changes that we see are by having that functional data. And you actually introduced the session by saying the other thing that we'd love to do, if we could do it again, would be to get at least some sampling from the lung compartment, because I think that's the thing that really would have enhanced our understanding of what's going on.
Thank you, James. I'm a big fan of multi-omics. And for the sake of time, we will take one more question from the audience, Dr. Kevin Harrell. And he has a question, do you know the extent of secondary bacterial infections, either community or hospital acquired, in your patient's population, particularly the non-recovered population?
Yeah, so Predict COVID-19 actually has kind of lots of arms and legs, and part of that included profiling bacterial co-infection or other co-infections in COVID-19. And so as far as we were able to tell, we didn't see an increase in secondary infections, but I know other authors have shown in longer term follow-up, so kind of 3 to 12 months, that patients who had severe COVID-19 in particular are more predisposed to other RA infections. And that is certainly something that our neutrophil data would indicate, that neutrophils are less able to deal with a further infection, and have indications of kind of lower ability to migrate over to tissues to be able to deal with those, and lower kind of bioenergetic capacity to be able to do their key functions. So it's something I think that longer term follow-up would be nice for as well. But, yeah, we didn't see that in our cohort.
Thank you.
Yeah, but that, I think, from a clinical point of view, what we see with the neutrophils is potentially important when you think about some places had real problems with post-COVID invasive fungal infections, for example, and that's very neutrophil dependent, the control of fungi. And when you look at our CD11b data, the migration data, and the metabolic data, you start to think, these neutrophils may well have an impaired ability to tackle these kinds of infections, and that might give us a clue to why we saw some significant problems in certain parts of the world. Fortunately, in this cohort, we didn't see it.
Great. Thank you so much, James and Merete, and for the great discussion. And so it's great pleasure to see such engaged group gathered to explore this high level of translational research for COVID-19. And the insights I shared truly highlight the depth and the diversity of perspectives we bring to understanding the proteomics analysis for respiratory diseases. Thank you all for the participation, and especially the authors and the panelists and the contribution to the discussion. Especially, thank you so much. And that's all for today. And Happy New Year.
Thank you. Bye-bye.
Bye-bye.
Thank you. Bye.
Thank you.
Thank you. Bye.